Well, welcome everyone. It's 8:01. It's time to begin grand rounds. And as always, I want to thank all of you for coming and spending time this morning to increase your knowledge about medicine. And I think it was like unusually slippery out in a weird way. So I think people are a little late coming today. So I have the honor today of introducing our grand round speaker, Nataliya Uboha, PhD, who is an assistant professor on the CHS track in our Department of Hematology and Oncology here at the Department of Medicine. And she definitely gets the award today for the best titled grand rounds we've had all year, Umbrellas and Baskets. So a little bit about Dr. Uboha. And I'm always bad with pronunciation, so I apologize. So Dr. Uboha has been trained at Yale University School of Medicine, where she did her MD PhD training. Her PhD training was in neuroscience. She then went on and did her internal medicine residency at Yale, and then did her hematology and oncology fellowship at the Simlow Cancer Center at Yale School of Medicine. We were very lucky to have her come to join us immediately after that here at University of Wisconsin in 2015. She has done some incredible things in a very short period of time here. She has 4 active grants, including 3 clinical trial grants, and I know more to come. And when I asked her what she's most proud of, it's really that independent work that she's doing in that area. And as you can see, like right in the front row are some of her peeps from GI cancer and some of her patients. So I'm glad to see you all moving to the front. That's fantastic. Dr. Uboha has 10 publications already in the short time that she's been with us here, and has been very active in important cancer groups. She is on the molecular tumor board here at University of Wisconsin, which is an incredibly powerful and important group. She is a member of the Eastern Cooperative Oncology Group for GI cancers. She is also a member of the GI Cancer Committee for the Big Ten Research Consortium, which we are very involved with here at University of Wisconsin. And she is on our Institutional Review Board Committee, which is another thing that takes a lot of time. We are incredibly lucky to have you and honored to have you come and give us grand round, so come on up.
Thank you very much for this very kind introduction. Good morning, everybody. It's a great honor to be here and to talk to you about some of the research we're doing. So here are my relevant disclosures. And I would like to start my talk by telling you about one of my patients. I'm not looking at buying time. I'm still hoping for cure. This was the last sentence in the email that my patient sent to me on 5/26/2017, just a few weeks after I met her. And this was just a few weeks after she transferred her care to mine after her primary oncologist has left our institution. So let me tell you a little bit about her. She was 61 at the time, and she was diagnosed with metastatic small cell GIST, gastrointestinal stromal tumor, in April of 2015. She had pretty extensive disease in the abdomen, so she had 2 debulking surgeries, which still left quite a bit of disease behind. And in May of 2017, right after we met her, she was starting 3rd-line systemic therapy, which was palliative in nature, which means there was no curative options at that time. She also had a lot of symptoms related to her disease, which were due to a large tumor that was present in her abdomen. So this is a picture of the CT scan. Shortly after that email, it showed it was about 20-pound tumor in her abdomen, displacing her organs. She had a lot of pain, bloating. She has ascites, which we were draining. She required frequent hospitalization because she couldn't eat. She also had debilitating lower extremity swelling, and she had a very hard time walking or even sitting. So of course that email caught me a little bit off guard because I tend to be very direct with my patients in clinic and talk about goals of care. And here I thought the expectations were unrealistic. But before even this email went out, I looked at her chart and realized that her tumor was never sequenced. So we sent tumor samples to different companies where they can analyze and see if there's any mutations specifically in the genes in the tumor that we can target with drugs. And so the following day, 5/27/2017, I got an email with these results, and that her tumor sequencing was positive for ETV-6 NTRK2 fusion. Who in the room, besides oncologists, have heard about this? Well, I didn't really either at that time point, so my plan was to look into it, but I didn't act upon it immediately. And so a few days later, I was at a big national conference sitting next to one of my colleagues, Dr. LeConte, actually, right there. And we were watching this presentation with the results of the drug that appeared to be too good to be true almost. And I said, oh my gosh, I think this is my patient who has this. And so I came back to UW, and even though my patient was greatly ill, worked very hard and tried to get her on a clinical trial for this drug. And she did start this drug in Chicago on a clinical trial. And she's had a remarkable response. A few months later, even a few weeks later, started to feel better. A few months later, her tumor really shrunk quite a bit. Since then, she transferred her care back to UW, and she's under my care now, receiving this drug. And right now, you can hardly see her disease on the scan at all. And so this drug is interesting. Larotrectinib is now approved by the FDA. It is the first drug that was ever developed and approved for a particular gene rather than a disease. And approval was based on combined data from 3 clinical trials and 55 patients total, both adults and pediatrics. So this is very unusual for the field of oncology, where we typically need randomized data, placebo controls, and large clinical trials. And so this experience has shaped my opinion of oncology, my practice, and my research in a number of ways. First of all, as a medical oncologist, I can't describe to you how amazing and rewarding it is to be able to provide this type of treatments to our patients. But it also stressed the importance of the research and clinical research, as well as preclinical research that we are doing, because really at the end, it's all about our patients and bringing these treatments to them. So during my talk today, I would like to talk to you, to share with you some of the recent advances in clinical research and oncology, and talk to you about the kind of clinical trials we are doing right now. So at the beginning of my talk, I will review some traditional clinical trial designs. Then we will talk about biomarker-based clinical trials using these traditional designs that we use in oncology. And I will talk about GI oncology and GI tumors, because that's what I treat in clinic. Then I will talk to you about the novel trial designs, basket and umbrella trials. That's what the title of the talk comes from. And then we'll talk about the promises and the challenges that these designs bring along with them. So we'll start about the traditional trial design. So historically, it has taken a very long time to get the drug from lab to patients. Overall, on average, it's about 14 years. And it's very high cost. And in oncology, it's only 5% of the drugs that enter phase 1 trials that actually get to clinic. So again, this kind of timeline would be completely not feasible for the patient of mine that I just presented. So what are the typical phases and designs in clinical trial that we see? Well, phase 1 trials tend to be usually the first in human trials. So the drug has demonstrated some promise in clinic, and then we try during phase 1 trials to figure out what is the right dose, and are there any limited toxicities for this dose when it's given to humans. Phase 2 trials tend to be a little larger. They're not randomized, but we're looking, again, at early efficacy signals, and typically further confirmations are needed. And phase 3 trials, which is kind of a gold standard, used to be gold standard in oncology for the drug to get approved, actually. It's a randomized trial. They tend to be quite large, a few hundred patients usually per arm, and we're comparing the experimental treatment to what is a standard practice in clinic. And so a few things, or one major thing, has changed in the last 15 years. In 2002, Human Genome Project was completed. Human DNA was sequenced. It took 12 years, and it took about $300 million to complete the sequencing. And since then, with the development of new technology, the cost for sequencing really fell off the cliff, as you can see on this green graph. And as that cost fell off, and this is the red graph, the red line on the next graph, people start to sequence more and more. And so that really affected, I think, the kind of drugs we're developing in oncology. So I want to walk you over this graph. So on the bottom, on the x-axis, you see approval year, and it goes from like 1940s to mid-2000s. And on the y-axis, you see the number of drugs that are approved per year. So let's follow the blue line. It used to be 1 to 2, maybe 0 drugs developed per year till about early 2000s. And if you move up to about 2011, 2012, there's a huge rise. And some years we have 6 or 7 new drugs that are FDA approved. Then let's follow the green line, which is the cytotoxic drugs, or some of my patients refer to poison. That line stayed flat, really. And so the line that really followed the development of new drugs in oncology is what we call targeted drugs. And when I say targeted drugs, I am talking about the drugs that specifically go under a particular alteration in the tumor cell. So whether it's a mutation in a kinase that really drives the proliferation of the tumors, or whether it's going after the receptor that's expressed in abnormally high amounts. So these are not the blunt tool that chemotherapy is. These drugs go after a particular change that is very specific to the tumor cell. So as we have learned more about tumor biology, we have also started to reclassify tumor. And so the paradigm has shifted in the way we develop drugs and the way we study oncology. In the past, it used to be all based on the primary location of the tumor. So the colon cancer patient would get this kind of chemotherapy, and then the pancreas cancer patient would get that kind of chemotherapy. And now, as we are learning more about molecular signature and these key alterations, we're really changing our approach, and it's becoming sometimes at least part of the puzzle, if not the whole puzzle, in our drug development. So the question is, are the traditional trial designs adequate in the current oncology market as we are learning more about tumor biology? And so let me give you a few examples where we actually applied our traditional clinical trial designs in patients with GI cancers. And I'm going to use biliary tract cancers as a setup to tell you this part of the story. So when we say biliary tract cancers, we really group a bunch of different diseases under the same umbrella. Biliary tract cancers include gallbladder, extrahepatic cholangiocarcinoma, intrahepatic cholangiocarcinoma. People with these diseases have different prognosis, these tumors have different behaviors, and they present differently, and yet we group them all together. And really, up until 2010, we did not have any standard treatments that were recommended for the biliary tract cancers. And we used to treat them similar to the way we treat pancreas cancer or sometimes small bowel cancer. The big phase 3 study was published in New England Journal of Medicine in 2010. And I don't want you to pay attention to all of the details, but basically what the study did, it grouped all of these cancers together, locally advanced, metastatic, and patients were randomized to either one drug, gemcitabine, or 2 chemo drugs. And it was a positive study. The overall survival for all comers in this trial was 11.7 months, and I want you to just remember the number. We'd like to say 1 year. This treatment, to this day, remains the standard treatment for patients with advanced biliary cancers. Okay? But again, it was like a very all-inclusive, very broad approach. So what has happened in the last 10 years? Well, we sequenced all the tumors, and as we sequenced the tumors and the tumor DNA, we realized that not all of these tumors are the same. Without even reading the very small print, you can see the gallbladder cancers have one set of mutations, extrahepatic cholangiocarcinoma, a different set of mutations, and intrahepatic, a different set of mutations. So these tumors are completely different on a molecular level. And it's not just an academic exercise. When we look closer at these mutations, we actually realize that we have drugs for a number of these mutations that are altered, and we have a potential in clinic to turn some of these mutations on and off, and hopefully can lead to some better outcomes. So let's talk about one of them, IDH1. It's a metabolic enzyme. It's dysregulated in multiple different diseases. In cancer, it's present in cholangiocarcinoma, up to 20%, but in other cancers, it's also present in great foglioblastoma, chondrosarcoma, in AML and MDS. And the interesting thing about IDH1 mutant AML and MDS, that there is already a drug on the market that's approved for IDH1 mutation in these diseases. So there's a drug we can study. So what does this pathway do? This is going to be in your CME questions, so please pay attention to this pathway. So IDH1 is, I'm kidding, it's the only joke. IDH1 converts isocitrate to alpha-ketoglutarate. Mutant IDH1 has a new function and actually can convert alpha-ketoglutarate to 2-hydroxyglutarate, which is an oncometabolite. And high levels of 2-hydroxyglutarate, which is present in cells that have IDH1 mutation, can then cause changes in DNA methylation, which ultimately results in changes in gene expression. And somehow the gene expressions that are altered lead to development of cancer. Exactly what genes are altered and the exact mechanism of these alterations remain unknown. So at UW, we opened a phase 3 trial. This was an international trial. Dr. Lubner was the PI, and we accrued really well for this trial. Again, this is a phase 3 trial, a ton of patients. It was randomized. So patients who already had chemotherapy and their disease progressed received either IDH1 inhibitor that's already on the market or were randomized to placebo. And at the time of progression, they were able to cross over to the study drug. And it was a hugely positive study. So on this graph, you see survival in months on the bottom line, and then progression-free survival probability on the y-axis. So if you look, let me see if I can point. Let's look at the 6 months time point. The red line is placebo. People who received placebo, basically 0 of them had disease that didn't progress, while people who were getting the drug had about 32% probability to be free of disease. So it was a very positive trial. We can't talk about survival advantages because people who were in placebo were able to get the drug, but it was great to be able to offer this trial to our patients. The drug was well tolerated. I had patients drive for hours just to be in my clinic to be able to have access to this drug. Let's talk about another alterations, FGFR2, which is fibroblast growth factor receptor, and it's altered in about 15% of patients with cholangiocarcinoma. Again, it's a receptor tyrosine kinase that's present on cell surface. There's 4 of them in the family, and typically is involved in the regulation of metabolism, endocrine functions, and wound repair. But when it's dysregulated, it can lead to cancer progression, oncogenesis, and drug resistance. And there's a few different ways this receptor can be dysregulated. And I just want to briefly go over this because I will be mentioning some of these terms later in the talk. The receptor can be mutated, and when it's mutated, it's constitutively active regardless of the ligand binding. The receptor gene could be fused to another portion of the protein, to a different protein, and when the receptor is fused to the protein, that fusion results in dimerization of the receptor, which is essential for its activation, and then the receptor is constitutively active. Or you could just have a ton of this receptor on the surface. It could be overexpressed, and when there are so many of them, the activity of the pathway is higher than normal. And so I would like to share with you the results of the phase 2 trial with an FGFR inhibitor. But before I do this, I need to give you a little bit of a tutorial about the waterfall plot, which is the frequent graphs that we use in oncology, and I'll be using these throughout the talk. So on the x-axis of these types of graphs that you'll be seeing, we have each bar represents an individual patient. On the y-axis, you can see the percent in tumor change from baseline. And so for us to say that somebody is responding, their tumor actually has to decrease by at least 30%. So that's the dotted line that you see. And if somebody's tumor is progressing, it has to increase by at least 20%. So basically, if you don't remember anything, these details later, you want the bars to be pointing down. If they're pointing down, that's good. If they're pointing up, that's bad for these graphs. So let's look in more details. FGFR2 inhibitor, phase 2 trial. It was not a randomized design. Patients already had at least 1 prior lines of therapy. And look, we are seeing overall survival of 21 months. So that's almost 2 years in patients who 40% of who already had 2 prior lines of therapy. I think this is a remarkable progress. And it's amazing to be able to give these types of drug to our patients in clinic. This is my patient that treated on one of these FGFR inhibitors. He was 63 at the time of diagnosis, and he was diagnosed with a large intrahepatic cholangiocarcinoma. It was not resectable because of its location. He received first-line standard chemotherapy with gemcitabine and cisplatin and progressed. And then he went on one of these FGFR inhibitor trials and had a remarkable response and was able to tolerate the treatment without significant issues, had a good quality of life. So I think, it's amazing to be able to bring these drugs via clinical trials to our patients in clinic. But let's look at the rest of the alterations. So this is a pie chart that kind of goes over the mutations we see in cholangiocarcinoma. In blue, unfortunately, is cholangiocarcinoma, biliary tract cancers where we don't find anything. Then you have another quarter of the pie chart, the red and the green, that have IDH1 or FGFR mutations, and then there's another quarter where you see small little slivers that are present in very small frequencies in these tumors. So it is just, when they're so rare, and now you're dealing with a rare disease with rare mutation, it's just not practical to conduct a large phase 3 trials. This issue is not limited to biliary tract cancers. As we are learning more about tumor biology, we are reclassifying even more common tumors. Lung cancer is now a collection of multiple different subtypes that we can define and target. And so as this is happening, and as we are reclassifying tumors, we are also changing the way we are conducting clinical trials. So these large histology-driven clinical trials are now being replaced with biomarker-driven trials, or at least biomarker is a big part of all of the clinical trials that we are doing. And so that brings me to the next portion of my talk, where I would like to introduce to you some of the novel clinical designs that we are now utilizing in oncology. And these are the large platform trials that are called umbrellas and baskets. So what are umbrellas and baskets? I will start with umbrellas. So an umbrella trial basically enrolls patients who have the same disease histologically. So all patients have lung cancer. But then the patients' tumors get profiled, and depending on what alterations they have, they will receive a different treatment. So patients with mutation A will receive drug A, patient with mutation B will receive drug B, and etc. So an example of an umbrella trial in cholangiocarcinoma and GI oncology is a BATCH trial. This trial is in development. I'm proud to say that Dr. Dusty Deming, one of my colleagues in GI oncology, is co-leading this development process. And this is a large trial that will be open throughout the country, and it's being developed through a cooperative group. And basically the idea behind this trial is for patients who have biliary tract cancers and are being treated with first-line chemotherapy to undergo molecular testing. And for these patients, at the time of their disease progression, these patients will then be assigned a particular treatment arm based on the molecular alterations we identify. So this trial is currently in development. So what are the advantages of umbrella trials? The advantages is that we are studying the activity of drugs and biomarkers within the context of the same disease. So you can attribute the results that you see more to the biomarker rather than disease biology. The designs are flexible. You can add, remove arm as more information becomes available preclinically. The disadvantages is that you need many drugs, some of which are tough to get. You need many tests. There are some biomarkers that can be tested with one platform as opposed to a different platform. And it can take a long time to accrue. So if you have a rare disease and a rare biomarker, one of those little cohorts may take a very long time to complete. So on the flip side, we have basket trials. So the basket trials enroll patients with different diseases, different malignancies based on the primary site. So you can have cholangiocarcinoma, you can have lung cancer, you can have prostate cancer, but they all need to have the same alterations in their in the tumor genome or biomarker expression. And so all of these patients, regardless of the histology, are then grouped together, put in the same basket, and they get the same treatment. So let's look back at the drawing board for cholangiocarcinoma. And let's look at the BRAF, which is present anywhere from 1 to 5% of cholangiocarcinomas, and it's this little blue sliver on the pie chart. Well, these patients with BRAF mutant cholangiocarcinoma were part of the large basket trial that enrolled any patients with almost any malignancy with BRAF alterations. There were 35 patients with biliary tract tumors enrolled in this trial. And for them to get 35 patients, they had to screen 700 biliary tract patients to find 35 with BRAF alterations. But the trial was largely positive. Again, waterfall plot, most of the bars are pointing down. That's good. Partial response was 36%, and overall survival was 12 months. Was almost a year, 12 months, which is quite remarkable because, again, these patients, a lot of them, 80% of them, had 2 prior lines of therapy. So we are moving in the right direction with our research. So what are the advantages and disadvantages? So the advantages is that theoretically it's a more efficient hit rate because you're trying to pool patients with different diseases. The frequency of these alterations differ, so you could accrue faster. Again, it's flexible design. You can you can alter the treatment as you go, and one test and one drug for all patients. The disadvantages is that from the basket, the interpretation of the results may be challenging because the results are preliminary and they can be affected by the tumor biology, as I will show you later. And so it's hard to know what to use as the right historical reference when we look at these results. And that brings me now to super baskets. And super baskets are the humongous trials. It's single trial infrastructure, and within a single trial infrastructure, you have multiple baskets depending on what alteration you're looking for and what treatment you can offer. And so these are basically multiple biomarker-based phase 2 trials under the umbrella of one trial infrastructure. So they have flexible designs, and you can add and remove arms as you go. And so the largest one of those super baskets that's open right now and has been ongoing for a few years is an NCI-MATCH trial. So this trial opened a few years back, I think about 4 years ago. Basically, any patients has a tumor that has progressed on standard line of therapy could have their tumor sequenced, and once their tumor sequenced, they could be assigned to treatments on one of many arms. I think there's up to 39 treatment arms right now. And so this is the largest precision medicine trial to date. In the first 2 years, 6,000 patients from across the country were screened and participated in this trial. It's open in every state in the United States. There's 1,100 participating sites, and we are still adding and developing more arms. And I'm very proud to say that Dr. Carrie Wasinski and Dr. Dusty Demings co-chaired and developed some of the arms, and Dr. Deming is still working on the new upcoming arms that will be added. So I've shown you some successes. Let's talk about some promises and challenges of this type of research. So there's probably more than this, but I tried to kind of organize my talk and concentrate on tumor biology and testing as well as infrastructure that's needed to conduct these trials. So I'll start out with the tumor biology that's very complicated and picking the right test to get patients on the trial. Let's look at this graph again. This is again the same waterfall plot that I've shown you with an FGFR inhibitor. Let's take a pessimistic view. So what about all these patients that did not have complete partial response, and what about those that progressed? Why did so many tumors fail to respond? And the simple answer and all-inclusive answer, well, tumor biology is complicated and the drugs are not perfect, so we need to work more of this. But let's try to dig a little bit more into this. So are we really targeting the driver of these mutations? So what do you mean when we say the driver? So when we look at mutations present in the tumor, we can classify them as driver mutations or passenger mutations. And when we look at when we get these sequencing results, sometimes we know if that mutation is, if that gene is mutated, that is definitely the driver mutation and that's driving oncogenesis. But sometimes the tumor just accumulates mutation because the cell is abnormal. It's really the mutation is present, but it's more of what we call a passenger mutation. It has no consequence. It's just really the result that the cell is abnormal. What makes it even more complicated, and then we have the variances of unknown significance and all of the compensatory pathways. And what I mean by variants of unknown significance, here we have the gene that tends to be the driver, but the mutation that's present in this gene is not the one they've seen before. So we have no idea whether that mutation is just like a polymorphism and it's just there, or whether that mutation actually affects the function of the gene. So this is just, so it makes it more challenging. And then, of course, compensatory pathways, because none of these mutations exist in isolation. There's multiple different signaling cascades. So it's hard to know what mutation in one gene does, but there's 10 other cascades that are mutated. We know that when we get the true driver, we can get results. And this is, when we found an NTRK fusion, we could go after it. So this is a beautiful waterfall plot with larotrectinib, the drug that my patient received. Everything is pointing down. Multiple different colors mean that every patient had a different tumor, and they all responded. Response rate was over 75%, and the response had lasted for a long time. At 6 months time point, 73% of patients were still responding. The problem is that this is a more likely scenario that we encounter. It's not as simple to find out who the driver is. I wanted to show you an example of the sequencing reports that we get in clinic. And so what happens, we talk to our molecular tumor board staff, Kayla or Hannah, and say, we would like to get our patient's tumor sequenced, and then we get an email a couple of days later, and the report comes out. And this is already a simplified report by, the company has done the sequencing, and there's a list of different mutations that are present and alterations, and then we have to decide, okay, we're going to do this. And so again, not straightforward. But these are two patients of mine again. One of them had FGFR mutation, patient A, and another one had an FGFR fusion. And actually, based on what we know, the fusions are much more sensitive, or tend to be more sensitive to FGFR inhibitors. And so both of these patients were eligible for a trial with FGFR inhibitors and enrolled, and one responded and one didn't. And so why? Well, it's complicated. There's a few different levels of complication here. First of all, tumors are heterogeneous. No two tumors among patients are the same, because there's a lot of different things at play, including immune system and coexisting mutations. And even inside the same tumor, different clones are present. And then when we look in the metastases, again, it's a combination of different clones, a different mutation that are different from original tumor. And then when you look at two different metastatic sites, now they're different. So it's complicated. It's challenging to target all of these. But also let's look further at two of these patients. Not only did they have different mutations, they also had tumors that originated in different sites. So again, the question comes back to where we started. So does histology still matter? So I would like to talk to you a little bit about although it's great to develop these drugs based on the biomarker and without in this histology agnostic way, we still have to know what we're dealing with. We still have to take into account histology. Let's step back to cell biology. EGFR signaling, epidermal growth factor receptor. This is an oversimplified slide, but epidermal growth factor receptors are present on cell surface and signal through multiple different pathways to the nucleus and can alter functions like cell survival and growth. BRAF is a serine threonine kinase that's positioned downstream of EGFR and is mutated in multiple different tumors. We have a drug that really can work quite effectively at turning turning this mutation off. And so we give this drug to patients with melanoma, and it works amazing. Everything is pointing down. So then we're excited because there's a subtype of colon cancer that also has the same mutation, and it's actually a bad player. So then we give this drug to colon cancer patients, and it doesn't work nearly as well. Why? Well, it turns out that in colon cancer cells, even though this kinase is mutated, EGFR signals very strongly and really is compensating for this BRAF mutation. It's really signaling quite strongly through these alternative pathways. And so you have to inhibit the EGFR and BRAF and maybe even something under BRAF to get the results. And so when we do that, when we learn more about the tumor biology within the context of the disease, we get better results. So basket 2.0. The histology agnostic, meaning any tumors can get with particular biomarker can get enrolled, but not histology blind. This is a trial that we also have open at UW. Dr. Burkhardt is the PI, and this is a trial for patients with HER2 mutant tumors. They all get a medication called niraparib. It's hard to pronounce all these names, but this is a tyrosine kinase inhibitor that targets HER2. Any tumor with HER2 mutation is eligible. However, depending on what tumor people have, we change the treatments a little bit. So patients who have breast cancer get niraparib plus two other medications. Bladder get niraparib plus paclitaxel, etc. So we take histology into account. And I think that's the next line of basket trials. The problem is that even if our strategy works, it's not forever. And resistance is basically inevitable with these drugs. These are the pictures from a couple more of my patients that I've treated with FGFR inhibitors in clinic. The first patient had an FGFR mutation, and she had a large tumor, intrahepatic, then responded, and then started to progress at some point during the course of her treatment. The second patient had an FGFR fusion, again, had a large tumor, responded, and then he started to develop satellite lesions, so progressed. So why does this happen? Well, there was an interesting paper that was published by Dr. Goyal from MGH. They have a rapid autopsy program there. So one of their patients on treatment with FGFR inhibitor passed away and consented to get the tumors sampled. So the patient's tumors subsequently, different metastatic sites were sampled and different metastatic sites were sent for analysis. And this is the results. This is a busy slide, but I'll walk you over this. So on the top you have a list of different metastatic sites that are analyzed. And you see liver met1, liver met4, so these are the sites that were analyzed. And on the y-axis, you have a list of mutations that were present. And I think without even going to colors, which some of them they demonstrate resistance or progression of particular sites, you can see that it's hard to find two columns that look alike. So every metastatic site was different. So what do we do with it? And actually, I saw the same thing in my clinic. So the first patient was rebiopsied, and we found very similar mutation to what they've seen before. The second patient had a liquid tumor biopsy done. So we have this new technology where we can actually isolate tumor DNA from blood. Tumors constantly shed DNA, tumor cells, there's a cellular turnover, and tumor constantly sheds their DNA into blood. And there's now new technology where you can send a tube of blood from a patient with cancer, and different companies can basically differentiate between tumor DNA and normal cell DNA, and then they can look for mutations in a set of genes that we typically go after with treatments. And so we're able to do this. The whole process takes about a week. We get the results quite quickly. So this patient actually had liquid biopsy done, and in the blood, 6 or 7 new mutations were identified. And so one way to look at this is very encouraging. What's the point? This is expensive. This is not going to work forever. But I think it's actually very important to understand what happens, to understand the timing of these mutations, and to learn more from them, because then we can model, as you know, why is this drug not working? How can we develop new drugs? And so some of these mutations, for example, prevented the drug to fit in from the pocket. So that could give us an opportunity to develop new and better drugs. But then the question arises, what should we test? You know, it's not reasonable to biopsy every single site. Should we just do liquid biopsies on everybody? At what time point? Should we biopsy the primary lesion? And the reality is we don't know. We don't know what to do yet, and we are learning. And again, this is where clinical trials come into play to give us information, to teach us how to use this information. But this is yet another publication that's coming from the same group on MGH. They had a patient with a BRAF mutant colon cancer. And the patient, as part of the standard of care, required resection of the brain lesion, it had a liver biopsy, then repeat liver biopsy, and also had a skin lesion that was resected. And on the biopsies of each of the lesions, so the solid, the traditional biopsy, all of the spots had a BRAF mutation, but they also had secondary mutation that developed during treatment. And none of the 2 lesions had all 4 of these mutations present. But when they said circulating tumor DNA, it actually had 4 mutations present. And so potentially it's more informative. Again, we're still learning how to use this. Let me give an example of how I've used liquid tumor biopsies in my clinic. So this is now the same patient I've shown you earlier who had great response to an FGFR inhibitor. He had FGFR fusion, went on FGFR inhibitor in March, had a great response in August, and then by October, started to develop small satellite lesions. He still had stable disease, so he could remain on treatment, but I was nervous that basically the next scan would show progression. So we decided to send his tumor DNA for sequencing, his liquid biopsy for sequencing, and indeed, in blood, he had multiple of these clones circulating, which we at that point knew basically predicts for resistance to the drug. So he had no disease elsewhere, so we decided to consolidate everything we could see with radiation therapy, since we basically knew resistance was inevitable. And so he remained off treatment from October to August of 2019, unfortunately developed a new site of disease, one peritoneal nodule at that point. We biopsied it, sequenced it. It was only a fusion, just like he had before. He did not have resistant alterations in that tumor, and so I was able to start him on another FGFR inhibitor that I obtained via compassionate use from the company. since we're learning more and we have more data with these drugs. And he's been on this drug feeling well and has had stable disease and stable tumor markers since August now. So what are the next steps? Well, I think we need to continue to learn about biology. Translational science is key. I also don't think that single agent inhibitors for these particular alterations, aside from NTRK, are going to be enough because the resistance develops, tumor biology is complicated. So really we need to think about combination strategies to tackle complex biology, whether it's in combination with immunotherapy drugs, with other targeted agents or radiation. We need to develop better drugs, and this work is not possible in isolation. We need to work as a team and to share data. For example, now, because I personally have had patients with 4 different FGFR inhibitors on trial. Again, rare alteration, rare diseases. And because of that, we have now formed a collaboration with Dr. Gayal from MGH. We are trying to pool our data together and really characterize better these patients with FGFR alterations. So just to give you a little preview of what we're doing here, some of the homegrown work. So this is the trial that I'm leading through the Big Ten Cancer Research Consortium. It's an investigator-initiated trial, multi-institutional, that will combine a drug like abemaciclib, which is a CDK4/6 inhibitor, cyclin-dependent kinase inhibitor, a targeted drug with immunotherapy. So that's one way to do combination strategy research. And another trial that's also we open here, it's a trial that looks at a combination of avelumab, which is immunotherapy drug, with radiation. Again, combination strategy for patients with esophageal cancer. Dr. Michael Bassetti is my co-principal investigator, and Dr. Dusty Deming is doing some elegant translational work in his lab to really help us learn why and why not the strategy may work. Let me bring you to the last portion of my talk, where we talk about how do you find the right patients, and it really does take a village to run these clinical trials. These trials are extremely complicated. There's new arms being added, there's amendments, there's regulatory work, there's IRBs, all kinds of things. And also the patients, it's hard to find, how do we get the right patient on these trials? How do we make sure that people know to refer patients to us for these trials? And so we formed, our village is the Basket Research Group that we formed in 2018. I'm very lucky to co-lead this group with Dr. Mark Burkhardt, and our group manager is John Collins, and there's a picture of some of these people are sitting here, and this is a great group of people to work with. So this is a multidisciplinary team that's dedicated to these biomarker and basket clinical trials. So before this group was formed, our research at UW and other institutions is typically done through the disease-oriented team. So I'm part of the GI group. There's a GU group. There's a thoracic group. And so if there's a trial comes for patients with lung cancer, definitely belongs in thoracic group. There's a trial that comes for patients with cholangiocarcinoma, comes to us. And so there was really no home for these phase 2 trials that were any HER2 mutant tumors. Nobody was really keen on having them through their group because it would enroll patients across different disease types. So you form the basket group, which is specifically dedicated to running these multi-cohort or disease-agnostic trials. So we launched 13 trials since 2018. We enrolled 55 patients. And we also piloted the just-in-time activation process for our patient population. And I'll tell you a little bit about what that is, but it typically takes 3 to 4 months to get the trial activated. So if a company or a colleague from another institution says we have a trial, we want to activate it, from the time we get the information says yes, we're going to do it, it's about 4 months, sometimes less, sometimes more, to get that trial to actually active and going at our institution, and really across from our partner institutions as well. And so we piloted this just-in-time process where we decided it's really hard to open a ton of different trials for rare patients. And so we said, how about we get the process? So if we identify a patient with rare mutations, then we open a trial. And so we were able to do that. And we were able to open a trial in less than 5 weeks. And I think that's a great accomplishment. And it's great to know that we are able to do this at our institution. So in the basket group, we were able to enroll patients to a number of different trials. Again, this is just like a sample of the trials that we have open for different molecular indications. Again, with FGFR inhibitors, we had 3 trials open. We have another one in activation. I've had patients myself on 4 different FGFR inhibitors. So how are we able to do it? So the essential components of this successful approval has been at least these three components are essential for successful accrual. So the basket dot, which provides the infrastructure for clinical trials. Dr. Mark Burkhardt also has spearheaded universal genomic testing. So he has led the partnership with Strata Oncology. We have research partnership with them. And so any patient with advanced tumors that comes and is seen in our institution can get their tumor profiled for free. And so all of my patients, my practice, get genetic testing. And I tell my patients, sometimes the likelihood of finding something is low, but it's zero unless we look. And I think my patient, the case I presented, is a great example of that. And we also have Precision Medicine Tumor Board, which is led by Dr. Dusty Deming and Dr. Mark Burkhardt, which is a statewide presence. So not only do we review cases from our own institution, oncologists from all around the state are able to submit these complicated reports like I've shown you, and our institution at no cost can provide recommendations of how to deal with these alterations, and also can make recommendations about clinical trial referrals. So in summary, I meant, and I hope I've shown you that the research with oncology agents have changed, and the new oncology agents that we are developing are not cytotoxics anymore. They are these targeted drugs. And it is important to study these drugs in a disease-agnostic manner, but it's not sufficient. We still do need to take histology into consideration. Tumor biology is way more complicated than one gene, one drug setup. And because of that, because tumor biology is so complicated, we need to continue performing ongoing translational research so we can advance our field forward. And also, it's not possible to do this work in isolation. So our collaborations that are ongoing and that we are developing are key. So UW is part of the ECOG Akron Cooperative Group. Dr. Deming and I co-lead the GI working group through Big Ten Cancer Consortium. We are part of Wisconsin Oncology Network, and we do trials with NCI. And I think putting all our efforts together is critical. Sharing data is critical. This work cannot be done in isolation. So, we haven't found cure yet. And at the end, this is all this work that I'm doing is for the patient. That's what matters, right? The reason we're doing all of this is so that I can go back to clinic and sit in front of somebody in the room and say, we have a better drug for you. And we haven't found cure. But I hope I've demonstrated to you that we are definitely moving the scale in the right direction. And that makes me hopeful. So I would like to thank to all of our patients and their loved ones, some of them are here in the room, for inspiration and for motivating me every day when I'm in clinic. But this is probably far from being an inclusive list, but I would like to thank all of the Carbone Cancer Center and faculty. I'm very proud to be part of the amazing group at UW. Dr. Regan and Dr. Bailey, thank you for their leadership and their support as my career unfolds. Dr. Deming is an amazing collaborator and colleague, and we do a lot of work together. Dr. Michael Bassetti is a radiation oncologist with whom I collaborate on some of the radiation oncology projects. Dr. Lubner, of course. I'm sorry I don't have that many jokes in my presentation. He is all around very positive and supportive and has helped with this talk. Dr. Mulcairn leads our GI group. Dr. LeConte has been very supportive and is part of my committee. And Dr. Mark Burkhardt, who's been absolutely instrumental to my career here and leads my faculty committee here. I would like to mention Dr. Howard Hochster, who was my mentor at Yale and now is a colleague and collaborator through Big Ten Cancer Research Consortium and through ECOG Network, and has been really an amazing force behind my oncology career. Thank you GI Group, Basket Dot, and I can't read the whole list, but I do have to mention the clinic staff, Stacy Baumeister and Jenna Wilkie, whom I work with clinic and who really help to take great care of the patients in clinic, but also allow me to have some time so I can prepare a grand round talk. And of course, this wouldn't be possible without my family. Thank you all.
Questions?
Thank you for that, Nataliya. Very good. In traditional clinical studies, your waterfall plot would also include control patients who would either receive standard therapy or placebo, and you could compare the outcomes in the two groups. And so I just wonder, where does control data and the application of controls to patients come in this model of treatment and study design that you have shown us here today?
I mean, that's a very good question. And sometimes when the signal is so...
repeat the
Oh, sorry.
Please repeat the question, yeah.
So these trials that I've shown do not have a control arm. So how do we really interpret these results without a control arm? And that's a great question. And I'm going to split into two answers. First of all, for drugs like larotrectinib, where we had 55 patients enrolled across 5 clinical trials, the signal was so strong that it was just not feasible to have a control arm. And so what happened with these drugs, and this is an isolated instance for this drug, the signal is so strong that FDA has approved the drug without the control arm, just because it's a breakthrough therapy, and again, you can't argue with the results that we are seeing. But with a lot of other drugs, what these trials are doing, they're still generating a hypothesis, right? The drugs are rarely approved based on the results of these trials. And so these are still phase 2 trials. And you're bringing up a good point, like we can't necessarily act upon these and not necessarily lead to the approval, but they definitely help us generate a hypothesis and potentially lead to the subsequent randomized trial design. Or in some situations, the drug can get much closer to the approval, and then confirmatory studies can happen after the approval. Since, unfortunately, it's exciting to see these results, in part because we have nothing else to offer for some of these patients. But the confirmatory studies sometimes do come after the approval.
Nataliya, thank you for your leadership, both of our group and of groups around the country in trying to find better answers. Can you point to how some of these clinical trial mechanisms overcome some of the sort of institutional inertia behind getting patients on clinical trials, and how can we get these trials better advertised to the community and to potentially other institutions?
That's a great question, and I think a big initiative that was done. How do you get, I think, knowledge about these trials to the community, and how do we get, how do we share this information with other institutions? I think individually, all of us in the Cancer Center work with multiple different oncologists in the community and have formed these personal relationships. But on a larger scale, though, as an institution, again, we are participating in a number of these networking groups, and I think that helps to bring the spotlight to our institution and also form collaborations. Through our molecular tumor boards, we have formed multiple research collaborations as well as relationships with the partners around the state. I am co-leading the Wisconsin Oncology Network as well, and we are trying to educate our partners and bring this awareness of these trials to the community. And there's also patient support groups. Our patients are online. They're part of the, one of my patients always comes to me with information from Cholangiocarinoma Foundation. So there's all of this information out there as well that we can direct patients to get information also.
So what about the future with precision medicine and designing individualized drugs for the patient? So we know the defects that the individual patients have based on the studies. When are we going to have that design of the drug for the patient rather than pulling something off the shelf or trying to try?
I think that's a little bit already happening with some of the immunology studies, like CAR T cells. When are we going to be able to design, I'll do my best to repeat this, when are we going to be able to truly individualize the therapy for our patients and design the drugs for the patients rather than pulling the drugs off the shelves that are already available there for a particular alteration? I think it's harder to do that with these targeted drugs that are going after a particular kinase, because we have the drugs that go after the kinase, and now we have to find out which patient's tumors are mutated in that manner. I think that individualized approach is already happening a little bit with immunotherapy drugs, like CAR T cell therapy, where you really try to learn something about patients' uniqueness of their tumor, and then you try to, in a way, program their immune system to attack these cells. this is still it's already used in liquid tumors, like hematologic oncology. It's still in early stages of development in cancer therapy, but there's a lot of interest in doing these types of trials, including cancer vaccines. Yeah.
Thank you. Excellent job.
Thank you. Thank you.